Thank you very much. Hi, everyone. I'm Giovanni. I'm a PhD student at the Stellenbosch University in the Center of Excellence for Invasion Biology, and my supervisor is John Measey. My co-supervisor is Sarah Davis, and I collaborated for this model with Kang Hui, with the professor Kang Hui. So this is the first chapter of my PhD thesis, and I would like to talk about an individual-based model that we built for understanding the invasive spread of a species that is currently invading Cape Town. So now just a little bit of background. So the guttural toad Sclerophis gutturalis is native in South Africa, as you can see from the distribution map, but is not native in the Western Cape. Unfortunately, in 2001, we observed for the first time some guttural toads in Cape Town, and more specifically in a suburb area called Constantia that is characterized by many private properties with garden ponds. So this species is able to breed using these garden ponds and is currently colonizing the area described by those red points. Because in Cape Town we have an endangered species that is very similar to this invasive species, the city of Cape Town decided to control the guttural toad since 2011. So they try to eradicate the species, capturing by hand as many toads as possible in these private gardens. The main limitation of this eradication is that we have access just to some ponds because we have to remove the species during the night. So this is a nocturnal species. So we have to go there physically and we have to remove all of them by hand. And most of these garden ponds are located in private properties. So you can understand that a lot of people are not super happy or very willing to allow access in these properties. So they started to eradicate the species in 2011, but after 5 years, we can easily see that the population is spreading. So the number of individuals and the invaded area is increasing over time. So we decided to study how the species is spreading across Cape Town and if this eradication is still feasible using an individual-based model. Now, the individual-based model is a very powerful approach because we can reconstruct the population dynamics of an invasive population using studying how the individuals are interacting each other and with their specific environment. So to build our model, the first step was to map using aerial images where all these garden ponds are located in Cape Town. So I literally checked thousands and thousands of aerial images, and I literally spied on people and in their gardens for locating all these different garden ponds. And unfortunately, in this invaded area that is described by these red stars, I counted more than 400 ponds in which the species can potentially reproduce. So I took the GPS coordinates for all of them, and I classified the size of this pond for building my model. In total, I counted more than 400 ponds, and then we construct the life cycle of the species within this pond network. Now, amphibians are characterized by a very complex life cycle in which we can observe 2 different phases, and terrestrial phase, in which we have adults, juveniles, and toadlets, and an aquatic phase in which we have eggs and tadpoles. So now, unfortunately, the literature about the species is very low, so I perform a literature review to estimate all these parameters. For example, we have the survival of adults, we have how many eggs they can lay, how many eggs each female can lay, and how many times per year, the survival of eggs, the survival of tadpoles, we have the survival of toadlets, the survival of juveniles, and how many of them are maturing every year. Now, I don't want to spend too much time on defining all these parameters, but there are just a few of them that are very, very important to understand the population dynamics of this invasive population. One of them is the tadpole survival. So this tadpole survival, in general, is characterized by 2 different components. The first one is the number of tadpoles, and the second one is the area of the pond. So these two different components are connected in this formula that you can understand easily, I'm pretty sure of that, in which we are describing a simple density-dependent dynamics. So when the number of tadpoles is very low, the survival of these tadpoles is very high. Conversely, when the number of tadpoles is very high, their survival is very low. This is happening because all these tadpoles are feeding on the surface of water, and they are competing for the same resources. So they are feeding on algae, and of course the numbers is shaping, and of course the area of this pond is shaping their survival. Something very similar is happening with metamorphs. When I'm talking about metamorphs, I'm talking about super tiny toads. They are less than 1 centimeter, and because they are so tiny, they are sensitive to dehydration. So in the first 3 or 4 or 5 weeks after metamorphosis, they have to stay within 5 meters from the pond. It also means that it's creating a very high competition, because all these tiny toadlets, and we are talking about thousands and thousands of them, are eating on the same insects. So in this situation, again, we have a typical density-dependent dynamics in which when we have a lot of metamorphs, of course, their survival is very low. But when we have just a few of them, the survival is very high. Then I want to speak about the terrestrial phase and how these terrestrial toads, like juveniles and adults, are dispersing across the pond network. Every year, they can colonize new ponds, of course. So we want to understand how a single individual is moving from one pond to another pond in 1 year. Of course, because we have a lot of different ponds, one specific adult can disperse across the pond network and can select so many different ponds all around. So the first step is just to calculate a distance matrix, so the distance between one target pond and all the other ponds. And to do that, the easiest thing is to calculate the Euclidean distance. So we have 2 ponds. For example, in this situation, we have 2 ponds with 2 different sizes. This one is small pond, and this one is a medium pond. And then we are just calculating the straight line between these 2 ponds. So for example, in which situation we are saying that the distance is 2,000 meters, 2 kilometers. But of course, this is not very realistic, because I mean, these animals, they are not moving following a straight line. So there are, of course, obstacles, there are barriers, and so we are not considering the landscape complexity at all. So we studied their behavior, how they are spreading, how they are dispersing, and we found that, interesting, for dispersing, they are using streets. So we are talking about a peri-urban area in which we have properties, gardens, walls, and streets. And we found that these toads are using streets for moving and from dispersing across the landscape and for moving from one pond to another. So to simulate this specific behavior, we use a technique that is called least cost path calculation. Every time that an individual is moving across the landscape, there is a specific cost of locomotion. Let's say in this situation that this toad is moving on the grass, the cost will be 2. If this toad is trying to go through this specific wall, the cost will be, of course, very high. But if this toad is jumping or like dispersing on the street, the cost will be 1. That is the least cost that we can have. So we created a raster in ArcGIS, and we built a model using these 3 different costs. And the software is just showing the least cost path distance between these 2 ponds, considering all these different costs. It's something that is happening, for example, in your navigator when we want to optimize, for example, the fuel consumption or something like this, or just the time that we should take for moving from here to here when there is a lot of traffic. It's exactly the same concept, but of course it's more natural than having just a straight line between these 2 points. So it means that from a toad perspective, the distance between these 2 ponds is not 2,000 meters, but it's 2,700 meters. So we calculated least cost pathways for all these different ponds and for all their combination. And then we use a dispersal kernel for understanding the probability that one toad has to move from one pond to another. To be clearer, if we are saying that there are like 1,000 meters between these two ponds, the dispersal kernel is suggesting that the probability for this toad to move from this pond to the second pond is 0.01. So the probability is just 1%. The dispersal kernel, as you can see, is characterized by a very long tail. So it means that most of these toads in 1 year are not moving more than 100 meters. So they are not moving much. But there are some atypical toads that are dispersing more than 8,000 meters, more than 8 kilometers in 1 year. Maybe you can think that this is a very big distance, but it's not, because, for example, I tested their locomotion in the lab, and I observed that just one toad can jump for more than 1 kilometer in just one night using a racetrack. I tested their locomotion, and I found that they can disperse for like 1 kilometer without stopping in just one night. So this is feasible, and some individuals can do that. And I want to focus my attention on this tail, because this is very important when we are modeling the spread of an invasive species. The behavior of these atypical individuals is so important for shaping their capacity to spread across the landscape. So after we built the model, and I validated the model using the eradication data that we collected from 2011 to 2015 in 5 years. So here we have some results. 2001, we observed for the first time in this pond 10 males and 10 females, more or less. In 2011, we started to eradicate the species, and I have data about the species in this area. So now we can see, performing a specific eradication, how the model is suggesting we will get some specific data. So we have two different scenarios. We have no eradication, in which we are not removing anything, or the current eradication, in which, in these red stars, we are removing tadpoles, adults, juveniles, a lot of toads, of course. So we want to understand if this specific eradication is effective and how is affecting the population dynamics of our species. So as you can see, we can have a demographic effect. The number of individuals, of course, that we are finding in the population is lower, but unfortunately, after 10 years, the species is everywhere. So what we are doing is just to reduce the number of individuals, but we can stop the spread of the species across the landscape, especially because, as you saw before, there are some individuals that are dispersing a lot and are driving the dispersal of the species, the spread of the species across the pond network. So now we tested a lot of different scenarios. For example, we can simulate that for those accessible ponds, we can try to dry all of them. So we are just removing water, and we are killing all these eggs and tadpoles. As you can see, we are getting a better result, but because we have access to just a subset of them, this is not enough for removing the population, for eradicating the population. We can also test a different scenario. We are just removing, in this situation, all adults from the accessible ponds. We are getting a better result, but unfortunately, this is not enough for eradicating the species. But as you can see here, it's not impossible to remove the species. We should have access to all these 415 ponds, and we should just remove a subset of adults and a subset of tadpoles without considering juveniles, metamorphs, and eggs, something like this. So here we have an accessibility problem. The access of these properties is very important and is affecting our capacity to remove the species in Cape Town. Maybe someone could suggest that a very smart way to control the species is to add like a sort of biological control. For example, we can assume that we are adding fish. So it's quite interesting because this species is characterized by eggs and tadpoles that are toxic. So many species of fish can feed on these toads, on these tadpoles and eggs. But there are, of course, some species of fish that can eat a lot of these tadpoles. So we can assume that maybe it's a good idea to add like a biological control in these ponds, and these fish, they will control the number of tadpoles. So we have a very basic scenario here in which we don't have any fish in these ponds. And now we are adding fish, so we can see what's going on. So here we are adding fish that are eating the 50% of these tadpoles. So it's a big amount of tadpoles, but as you can see, this is the total number of adults in our population. As you can see, we are not getting any result. So the result here is that we are not getting any result because the survival of metamorphs and tadpoles is density dependent. So it's density dependent. So we are removing tadpoles, but we are at the same time increasing the survival. So we are not getting any demographic effect. Maybe these fish, they are not eating enough, so we can eat fish that are eating more. So here we are adding fish that are eating the 90% of tadpoles, but we are getting exactly the opposite result. So the number of adults will be higher and not lower. So this is a very negative biological control. Then we can add fish that are eating the 98% of tadpoles. As you can see, also in this situation, there's no difference. Otherwise, if we are adding fish that are eating all these tadpoles, so it's like, for example, drying these ponds, we are getting a result. So the advice here is that if we want to introduce like a biological control, if you want to use a biological control, we have to consider like non-linear dynamics in population dynamics, in this kind of population dynamics. My conclusions are that the current eradication strategy will be ineffective in the long term, especially in 10 or 20 years. And increasing the number of accessible ponds, it is still possible to eradicate the species. The best strategy so far is to remove adults. And we have also to consider that management of invasive species characterized by complex life cycles, like amphibians, requires to take into account density-dependent survival and non-linear demographic dynamics. So, thank you very much for your attention.
Thanks for the talk. There is time for some questions. Mark?
Can you say more about how you parameterized your dispersal function?
Sure. So the dispersal was calculating by different people using a mark recapture study. So unfortunately, we don't know anything about the dispersal of this species in Cape Town or in general in South Africa. And we don't know much about the frog dispersal in general. So I use a dispersal kernel that people estimated in Bufo Fowleri in the States. So I use exactly the same dispersal kernel, and unfortunately it's just an assumption, but I did the same with some demographic parameters because unfortunately the literature about this species is very limited. But I think it's quite accurate because in general toads are characterized by some event of long dispersal. So some of them can disperse a lot, as I observed, for example, testing their physiology in the lab. So, good question. Thanks.
There is time for another question.
Okay, so I'm trying to look at the biological control where you introduce the fish and you see that there is no effect, and what was expected from the fish was to eat the frogs, right?
Yeah. Because,
Could it happen that since the frog, they disperse, and maybe the fish will remain in wherever there is water, in the pond that there is water, and not find anything to eat? And have you looked at that? What is the interaction of this new entity that they have introduced in the environment?
Okay. Now that's a very good point. I mean, that was just a very theoretical scenario for saying that you can control the number of tadpoles, but you can get like an opposite result.
No, but do you think it's right to draw such a conclusion just from a toy perspective like that?
Like
without really bringing in the
yeah. I mean,
fish into
you're perfectly fine.
consideration.
So that
Maybe even the life cycle. Maybe fish were dying before, were compared to the time of the reproduction of the frog, or their prey. Maybe.
yeah.
Because
That is just for
trying
seeing.
to see at least some reduction, even if their efficiency is very low, but their
No.
presence as predators is
Not-
supposed to really
okay, yeah.
show some effect at any level of the model.
Yeah, that's a good point. So we have, so here I simulated that I added fish just in these accessible ponds. So it's just a subset of ponds in which I'm adding fish. But at the same time, when you are adding these fish and they are consuming these tadpoles, when you are checking the first graph in which you have the density dependence relationship between like number of tadpoles and the day survival, you can easily see that when you have just a few tadpoles in your pond, some of them are able to escape from the fish, and all of them are surviving. Because if you have just a few of tadpoles, you will have also a few of metamorphs outside the water. They are not competing each other because the density is very low, and most of them are surviving. So it's very counterintuitive result, but this is called Hydra Effect in a lot of different populations, in which they try to manage a population using a biological control, but they call it Hydra Effect because you are cutting some head, and new heads are coming out. So you are getting exactly the opposite result because you are increasing the survivorship of them. I don't know if I answered your question, but we can discuss later if you want.
Okay.
Yeah.
So Giovanni's given you one aspect of invasive species and frogs, which is that we can have invasive frogs, and I'm going to speak from the other viewpoint, which is that invasive species can affect native frogs. So South Africa, as some of you may be aware, has an amazing diversity of frogs. Here's a very large bullfrog that you'd need 2 hands to pick up the male sitting on top, but maybe only 1 hand to pick up the female. And this has a very large distribution in southern Africa, so many of you may be familiar with it. Down to, we have some very small species as well. This is called a micro frog. It's actually critically endangered, and it has a micro distribution. So it has these 3 very disjunct distributions, and this one is right in the middle of the last piece of sandplain fynbos, which is residing in the middle of Kenilworth Racetrack. So very small distribution there. And in general, when we look at the distribution of the threatened frogs, what we see is that they're in these coastal areas, and there's one place in particular where there are lots of threatened species, and that's right here on the Cape Peninsula where we are now. A famous herpetologist called J.C. Poynton pointed out that the Cape frogs in particular are very special, and that's been seen now with much more global studies. When they looked at amphibians, you can see that there's actually a big species turnover when you go from the Cape right throughout the rest of Africa. So this is a very special place, and the reason we think that this is a special place for frogs is we have this rather strange winter rainfall regime here in the southwest of Africa, whereas elsewhere we have summer rainfall. There's the winter rainfall where we are right now, and some people were complaining about this at lunchtime, and summer rainfall that many of you may be more familiar with up country. So, as I said, we have a great diversity of frogs in southern Africa. There's 12 families, but only 6 families here in the Cape Peninsula. But those 6 families unfortunately have lots of threatened species. And when we look at the reasons for the threat, so you look at the distribution, you can see a lot of these are coastal species. They coincide with places where people live. There's also upland species, And a reason for a lot of these threats are invasive plants. Okay, so invasive plants go into mountainous areas and cover large tracts of land. One of the things they do is suck up water, and this is what's needed by a lot of these frogs. So one of the things that we've done, or is being done, in order to keep an eye on this, this is a man called Andrew Turner, whose name was at the beginning. He works for Cape Nature, and he is monitoring a population of frogs here in Grootwindhoek, where this circle is. And you can see that this very special technique that Andrew is using to monitor the frogs is putting his hands behind his ears and listening for the frogs. And what he's attempting to do, yeah, there are no frogs calling here. What he's attempting to do is count the number of individual frogs, which you can do if you have between 1 and 10 frogs. If you have more than 10, then you can estimate, well, maybe it's between 10 and 20. If you have more than 20, you can probably say it's less than 50. And otherwise, you're somewhere between 50 and hundreds of frogs. You're not quite sure. And I spent some time with Andrew watching him monitor frogs because we were writing a chapter of a book together about monitoring, and I thought that there must be a better way of doing this. There should be some better way. And lo and behold, I came across a publication about ovenbirds in Australia and getting some density of the birds through acoustic signals. And I said to a colleague of mine, I was working in Sandby, where Sabrina works at the time, a colleague of mine, Res Altveg, who now works at UCT, I was saying, Res, can't we do something like this with the frogs? Res said to me, I'm off to Scotland to go to a meeting, and there's a guy called David Borchers who has visited Ames, I think at the beginning of this year. David gave a course at Ames. And he said, well, I'll ask David about this technique because I know that he's involved in these kinds of models. And together, David visited Cape Town. It turns out that David's South African and was looking for reasons to come and visit Cape Town. And we put together a grant application for National Geographic to work on various species of very small frog that live in mountainous areas. One of the things that all of these small frogs have in common is that they're affected by the spread of alien plants, and in particular pines, Pinus radiata, I believe. Here you can see Andrew. This is another site of this critically endangered species, Arthroleptella rugosa, and the whole of this patch that has been recently burnt is invaded by pines, and you can see the pines going up the slopes here as well. For those of you who know the area, this is just above Caledon in the Klein-Swartberg. So the technique that we were interested in using is something called spatially explicit capture-recapture. Can I ask right now whether anyone knows anything about spatially explicit capture-recapture? I can see one hand has gone up. Oh, there's a hand sort of waving in the air a little bit. Okay. This is a special grid that I constructed for those of you who have slight stigmatism. You might see the slight wavy lines going on here. But the guy who invented spatially explicit capture-recapture, a man called Murray Efford in New Zealand, was eradicating rats from some of New Zealand islands. And he put out a regular grid of pitfall traps for the rats or mice. I think sometimes they were mice to fall into. But he noticed that he was also capturing threatened New Zealand skinks. And what he would find is that he could capture a skink in a trap and he could mark it. And next time he came along, that skink may be found in another trap. and so, oh, he's noticed that he's found that skink before, but then next time he came along and he found some rats, but he found the skink again in another trap. And what Murray realized was that normal capture-recapture data actually doesn't take any notice of where any of these traps are. But clearly that there's something about this skink that has a home range in this area, and that it's more likely to be caught over here than it is somewhere over here. Here's another skink, another couple of skinks. So you can see that this one was captured several times here, and this one was just captured a couple of times in the middle. So the idea of spatially explicit capture-recapture is that you can use this information about where you're capturing the individuals to give you some idea not just of the number of individuals you have, but also the area that these individuals occur in. So for those of you who are used to capture-recapture, what you end up with is a population estimate, amongst other things. You can estimate all sorts of parameters, but you have a population estimate in space, but you don't know where the limit to that estimate comes from. What you get with the spatially explicit capture-recapture is that you get a density estimate. So you get an estimate of individuals within a certain amount of space. So as I say, this was started in a paper by Murray Efford and has now become the mainstay of many ecologists, and in particular, people are using camera traps. So there's lots of people who put up camera traps and just to look for leopards in this kind of area, well, not right here, but in the mountains near here, and because they can individually recognize the leopards and they spatially know where the camera traps are, they can estimate the density of leopards within a particular area. Now, it turns out that you can extend this technique to not only physically capturing and recapturing animals, or photographically capturing and recapturing them, but also you can do it with sound. So you can do something called acoustic spatially explicit capture-recapture. So imagine that these 6 crosses are microphones, and that here we have a number of frogs in an area. You can't see the frogs. I'll show you why in a minute. The first reason why is because they're very small. They're about the size of Giovanni's metamorphs. This is a microphone here, normal size microphone I had, with a little adult male frog sitting on top. So each one of these microphones itself has a detection probability function. So there's an area outside the microphone that it can't hear, but equally there's an area quite close to the microphone where the detection probability function is actually very high. And when you have 6 microphones, you can draw this detection probability as a function for all 6 of them, and you get this rather large plateau type diagram that was produced for me yesterday, I'm happy to say. So there we have an area now somewhere like this around these microphones where we can be pretty sure that we're going to hear frogs calling with a very high probability. This is more where I'm implicated in this sort of arrangement. This is actually the mountain right above us now. It's Muizenberg Mountain, or Plateau. There's a recording unit sitting right there, and each of these 1, 2, 3, 4, 5, 6 microphones is sitting on 1.5 meter high pole. So you can see that's the sort of thing that it looks like. Each one of the microphones is recording calls coming from the frogs. So there's the frog, that's what its call looks like, and we run that through an automated call recognition piece of software. And let me say at that point that there's all sorts of avenues for investigation in terms of recognition of sound. And I think there's a student currently at Ames looking at whether we can get individual recognition of calls rather than what we're doing at the moment is just recognizing the species from all sorts of other things that are calling. So the output that we get from that automated call recognition software will tell us which of the microphones is hearing a call. It will also tell us the amplitude or the signal strength of the call. And what else will it tell us? Oh, it will tell us the start time of the call. Okay, so we get those three pieces of information. So using our spatially explicit capture recapture, if we use that formula, we get this probability distribution function of where the frog is most likely to occur here on the periphery of the area. If instead we use the start time of the calls, we get a kind of triangulation piece of information, which is actually far more accurate, telling us where the frog is likely to occur. And we can also use the signal strength or the amplitude, how loud the call is on each one of these microphones. And that also gives us a probability distribution function. And then the rather natty piece that I think is very special, but the statisticians tell me it's not particularly special, is that you can actually add these three things together. And you can produce a rather tight distribution where the frog is most likely to have been calling from. Excuse me. Here's another example. This time, the frog is heard on all 6 microphones. So our spatially explicit capture recapture is just going to tell us that it's in the middle somewhere. But our time of arrival information gives us a very tight area where it may be heard. Signal strength, a little less. Signal strength, of course, if the microphones are deposited in areas of dense vegetation, we could have interference between the microphone and the frog, so we're not quite sure where it is. So these tend to be a bit looser, but they do add to the information of where the animals are captured when you add all 3 together, and you can see there's a actually tighter probability distribution function there. Dave made all of us put some equations in. Sabrina and everyone else has gone through theirs, but I'm not going to try right now. Yes?
Yeah, please. I want to know how good is a microphone to differentiate the frog from other animals?
Okay, so the microphone isn't what's differentiating between the frog and everything else. What's doing that is this software. So we use a software called PAMGuard. There's actually lots and lots of software out there, and this one's a free bit of software that recognizes calls. So that this is what is recognizing the call information. The particular minute of calls that we use for this presentation has lots of songbirds. You can hear insects buzzing by and all sorts of other things. But it's this software that is picking out just the frog calls and then ignoring everything else. Does that answer your question?
Yeah, so somehow this is a little bit different than classical capture recapture, because rather than a single individual being caught sequentially in different traps in time, it's a single individual maybe being caught by all traps at the same time or by a subset of traps at the same time.
So it turns out that you don't need the initial capture in order to put the capture history of something into capture recapture. Does that make sense? No.
Yeah, we talk about it later.
Yeah.
It's intriguing, but I don't quite see the connection.
Okay. So yes, that's an important point in order to jump from physically capturing to auditorily capturing, if that's a word, I'm not sure. Okay, so there are lots of equations involved, and the man who produced those equations is Ben Stevenson. We've got a paper that goes through them in some detail, and we've got another one that's in review at the moment that talks more about the methodology. So as I mentioned earlier, the process actually gives us an area to which we're listening in order that we estimate the number of animals within this particular area. So even though I showed you that the spatially explicit capture recapture wasn't giving the best estimates of where the animals were calling from, it's important in actually generating this area that we're actually listening to in order to give us density. Without that, would actually be a step backwards. So what can you do with this kind of information? Well, we had an honour student a few years back at UCT who used repeated sampling at 3 sites to look at how call phonology, or the number of animals calling, changes throughout the year. So these frogs call an awful lot during time, from May all the way up to November. And you can see that each of the 3 populations we monitored actually has a change in the way they call throughout the year, with a sort of peak calling period in July or mid-July. One of the unexpected findings of the study was that each time we put the recording apparatus out, we actually were sampling a slightly different area. So this 00 is based on microphone number 1, and most of the time we got about 20 meters either side of the microphone, but on occasion we got beyond 30 meters outside of the microphones. And this means that, so it could possibly be that the batteries in the microphone were going down and down and down all the time. But that wouldn't explain why most of them, they just kind of carried on indefinitely. That's what I thought at first. But it's probably more likely that different atmospheric conditions are better at conducting the calls, or that the vegetation that is on the ground here in different levels of very slight wind has different effects on being able to hear different distances. The reality is we don't know why these distances are varying so much, and this is one of the 3 sites. The other sites were actually a little bit more variable. But for our method, if you can imagine that we're looking at the density of frogs in the area, it's not actually important, because the density will always be the density of frogs in a particular area. Other methods, people who are just using single microphones or a stereo system, have no idea of the area they're actually sampling. And so if it's changing a lot all the time, they're not really aware of what they're sampling. So this was an unintended consequence of our study that turns out to be quite important in acoustics. Another unintentional consequence of the study that the statisticians just said, yeah, but of course, is that you can actually localize the frogs and where they're calling from. So you can get a lot of information about the calling ecology of the species. You'll notice that it looks like you're getting some kind of interference pattern towards the edge, but don't forget that around here is the limit of your probability density function, and so actually the localization of these animals towards the edge is actually much, much worse than it is towards the center of the microphones. So it looks like there's a pattern, and that's because you can imagine this being some kind of lens blurring as you get away from the center. So what can you do with that? Well, Here's our Pinus radiata plantation with lots of invasive individuals around the edge getting into the felt. And what we can do, and we have a student currently starting fieldwork doing this, is to look at what effects these alien and invasive plants have on frog populations. So we can actually find out, because we know that we can hear these frogs in these plantations, but we don't know whether the densities are increased or not. I'm using the call localization function. We can actually go right down to a much smaller level, and we can find out whether an individual pine tree is actually avoided by individuals because it's having some sort of impact on their microhabitat, on the micro-distribution of the animals. Okay, thanks very much.
Thank you very much. There is some time for questions. So please.
I'm looking at the calls that are made by the frogs. There are insects that also make similar calls. How do you discern this within the microphone?
So again, as I say, it's not the microphone that's doing that. So the microphones sit out in the field. We put them out for about 40 minutes. We record simultaneously from all 6. We then take the recording back to the lab. In the lab, we run the recording through this software, which is called PAMGuard. Into PAMGuard, we put a formula, if you like, where we say we're interested in calls between this and this amplitude that occur over this and this length of time. And this is called a click detection function. So we program PAMGuard to say we're only interested in these frogs, and then we validate that by listening to minutes and minutes of calls against things like birds and insects to make sure that it's not picking up any other species. Birds are giving us particular problems because they have rising and falling calls, so we have to make our box very conservative. The statisticians tell me that it's important that we get no false positives. False negatives are not a problem, but false positives are a particular problem for this technique. So we actually make that box very tight.
So distance is also part of this in the analysis. And for that reason, do you think Konoko would be one of the softwares to analyze such a kind of data?
No, I wouldn't use Kaneko. So to do the spatially explicit capture recapture, which includes the signal strength and time of arrival, Ben Stevenson has written a piece of R code called AMBSECR, which is available from GitHub, I think. But the spatial, so you're right that the spatial side of it is very important, and we're exploring that at the moment. So we need to know the relationship, the spatial relationship exactly between the microphones. So we measure to the nearest millimeter how far the microphones are from each other. So we have the exact spatial coordinates for each of the microphones. And what we're trying to do now, using a drone, is to map that area in terms of the invasive plants and things like plaths and rocks and other micro distributions within that acoustic area, so that we can see what effects all these things have on the frogs and where they choose to call.
Is there an optimal spatial sampling design for the microphones?
Yeah, so that's an interesting question. I think the answer is yes, and the conclusion we've come to so far is that we don't care. Let me try and explain why. So back here I showed you this, oops, gosh. Yeah, so I showed you this, which clearly has a flat top across the top of this detection probability. So what you could do is you can keep moving each of these microphones further away until you stretch the possibility of this detection probability as thinly as you can across the top without it starting to dimple. So you can imagine the further that these are moved away, you're going to have curves or domes at the top, around the tops of each microphone. So the ultimate would be to move them far enough apart, but before you really present any of these undulations occurring. But because we don't understand why this is happening, if we did that, so if we stretch them to their limits, then we may just get some gust of breeze or gust of wind coming through that completely changes this, and then the dip would be so great that it would just render the technique not usable for that particular minute when that occurred. So I should say that each one of these lines here represents a minute subsample from a half-hour recording. And we do, I think there's 10 subsamples per recording, so this is lots and lots of subsamples of lots of recordings. Does that answer your question?
Okay, thank you. I'm worried about two things here in this microphone. What if we are having, let's say, 3 or 4 frogs standing near in one microphone?
Mm-hmm.
Now, how you distinguish that was 1 frog or 3 frogs standing near the microphone?
So as long as the animals are close to the microphones, you don't actually have a problem, because we're having sub-centimeter accuracy on localizing calls. So it's really very tight calling. One of the things that a lot of people say is, what happens if the frogs call at the same time? Which is this kind of different part of the same question. And the answer to that is that the recorder records at 48 kilohertz. So that's 48,000 times a second. We have a possibility of getting a different call. So the possibility that the frogs, different individuals, would synchronize their calls to that level of accuracy is quite low. That's what gives us the time of arrival accuracy here as well. And the other advantage we have is that these frogs, and many frogs in general, will specialize in alternating their calls. So they deliberately won't call at the same time as a frog that's close to them, because that would mask their call, and they want their call to be heard differently. So that's part of the biology that actually helps as to our advantage in recording them.
Okay. And also the distance. Let's say you're talking about the distance of the frog and then the microwave. At what particular distance where the sensor is sensing the call, like from the microwave?
Yeah, so...
Maybe you can repeat the question, because I think...
Sorry.
Yeah,
So
the question.
The question, as I understand it, is what happens as the frog gets further away from the microphone. So that is more or less covered here. So this is the detection probability. So each microphone has a detection probability function. And as the frog moves away, if you like, from the microphone, it has a reduced detection probability.
I'm concerned about the distance, actually. Did you consider the distance, let's say, at this particular position, it's whereby you sense the call? Do you get my question?
So you're saying what?
The distance from the call to the microwave, actually. At what particular position or distance where you say, now I'm starting to record the call from the frog.
Okay, but that's captured by this detection probability of all 6 microphones. So this is distance on the X and Z axis here, and its detection probability, sorry I should have said that earlier, is from 0 to 1. So this is actually a distance here, and this distance across the bottom here is about 50 meters. And I think it should be labeled on these axes. These ones. So yeah, it's 40 meters all the way across here. And this is that same detection probability, but it's cut off here, I think, at 0.95.
Yeah, I just wanted to know if you have also considered other ecological factors, maybe at micro-habitat level, I don't know, for the conservation purposes, because maybe you'll be able to predict the spatial distribution in addition to what you have mentioned, that they are seasonal and they are also related to these invasive species. Maybe soil moisture levels, soil structure, or soil texture factors.
So the question is, are there any other factors that could influence the distribution, the micro-distribution of the frogs within these areas? And of course the answer is yes and no. That we don't know what they are yet because we've only just started to investigate. One of the things that I first noticed when I looked at this particular print was that there's actually a stream that runs through this site like this, and that it seems that the frogs were congregating along the edges of the stream. And of course it would make sense that they're not sitting right in the middle because they'd be washed away. But one of the interesting things about this species is that when you walk into a chorus, so they're sitting on the ground in sort of wet areas, when you walk into a chorus you can't really even hear yourself think, let alone determine how many frogs or where they're calling from. So it is going to give us all sorts of information about the micro distribution. And once we've started investigating, well, maybe in 2 years' time I can answer your question with more accuracy. Okay.